Okay, so let's go ahead and begin. So again, we're really thrilled to hear from Dr. Fanous over research on serious mental illness. So he is a professor and chair of Department of Psychiatry at University of Arizona College of Medicine in Phoenix. And prior to coming to University of Arizona, he was the chair of psychiatry at SUNY Downstate Medical Center for 5 years. He is a clinician, scientist, educator, and leader who has distinguished himself in the field of psychiatric uh, genomics research. And for over 20 years, he has applied linkage, candidate gene, resequencing, whatever it is, genetics wide association, next gen sequencing methods to uncover genetic factors, risk factors for psychiatric illness. Um, he's made he's been deeply involved in a lot of impactful initiatives in the field over the last 15 years, which have led to publications in Nature, which is a really hard magazine to get articles published in, and other high impact journals. And he has a whole list of the research that he's done. He's leading the phenotype analysis of the genomic psychiatry cohort for both bipolar and schizophrenia. We're really excited to learn about how we're entering into a new age with AI and genetic sequencing that I think will finally maybe lead to some breakthrough um, cures or better medicines for serious mental illness. So we couldn't be happier to have you tell us what's going on. And before we begin, do you prefer speaking and then having people wait till the end, or do you prefer people raising their hand and asking you questions as you go, or what's your preference?
Well, I mean, it really depends on the audience's preference. This is a scientific talk, so I realize that it will be technical at some points. I mean, I'll try my best to sort of break it down and make it sort of understandable to people who don't do research, and that includes many physicians, of course, as well. If people would like to ask questions, if that's something people are more comfortable with, I'd certainly be happy to do that during the talk.
I think it may be, because I'd hate for someone to go through the whole thing and not know a specific term and then kind of miss out on the connections throughout the talk. So
That's fine. Can you see my screen?
we can.
Okay, so I'm going to go ahead and just go to slideshow. So can you, hopefully you can see my first slide now.
Yes.
Okay, great. And you can see my pointer hopefully as well,
Yes.
because I'll be using it during the talk. Okay, perfect. Thank you so much for the introduction, Ms. Goldstein. I really appreciate being invited. It's a real honor to speak to this organization. I've only recently gotten to know this organization at the recent event that was held at Wrigley Mansion. I was very pleased to be invited to that. I am an academic and a scientist, but I'm also a clinician. I've been seeing patients for decades, starting in New York City, where I did my training at Bellevue back in the 90s. I've worked in Virginia as well, in the Washington, D.C. VA Medical Center. I've worked in the Veterans Administration system for many years. I also have people in the family with illness as well, so I've seen things from both sides of the fence, so to speak. And I want to congratulate your organization on what you've done. Coming together to really do some tremendous things to make life better for some of our most vulnerable and truly ill individuals, people who oftentimes will never get a chance without organizations like yours. So I want to thank you on their behalf and congratulate you on the fine work that you're doing, and encourage you to please keep up the great work, because it's absolutely fundamental to the well-being of thousands of people, as you very well know. So what I thought I would do today is really bring to this audience an update on the genetics of schizophrenia. So I'm going to focus on schizophrenia rather than bipolar disorder and major depression today, partly because it's a more severe illness, and as many of you know. but also because we've had more progress in schizophrenia genetics than we've had in the other disorders for various reasons. We don't need to go into all of them. But I'm going to be focusing on schizophrenia. Now, as I was mentioning earlier, science by its nature is very technical. So, part of what I aim to do is bring to you some of the latest findings in the science. But I do realize that in order to express some of these ideas and things like that, I do have to use technical language, and that may not be something that people have, at least these technical terms are things some people have not heard before. So I'll try to break it down a little bit to the best of my ability. But again, I will be happy to do that even more so if needed. So we can go ahead and take questions to some extent during the talk. Okay, so the title of the talk is an update on the genetics of schizophrenia, the road to precision psychiatry. I have no financial disclosures. And before I go into the meat and potatoes of the talk, I actually like to give my list of collaborators ahead of time, just in case, because sometimes in the past I have forgotten when I've had it at the very end, and I don't like to not give people credit. The work that I'll be presenting here is highly collaborative. I mean, it could not be done other than through the collaboration of hundreds, sometimes thousands of people, many playing small parts, some playing very big parts. These are my main collaborators. As I mentioned earlier, I was in Virginia, so I started out at Virginia Commonwealth University, and I've collaborated with people at many universities, which is really the rule rather than the exception in our field, because it is such a very broad area where lots of people from many different modalities and areas of specialization have to come together. So my list of collaborators is huge. These are my main collaborators, and I'm just going to just very briefly show you this slide very quickly, which is a list of the co-authors on our most recent paper. Of course, we can't go into every single one and what they've done.
So it takes a village, right?
Absolutely. It takes more than a village. In some cases, it takes a megalopolis, let's say. Okay, so I thought before we go into a lot of the technical details of genetics, let's talk about what schizophrenia actually is. There's, we think of schizophrenia today as a certain DSM-5 or diagnosis, ICD-10 diagnosis, but it's important to remember that this diagnostic, framework that we have today has evolved over many, many years. And what we think of as schizophrenia today, we didn't always think of it the way we conceptualize it today. So going back to the mid 19th century, there was no concept of schizophrenia. There were many psychotic rubrics, so to speak. So they had concepts, for example, such as hebephrenia, paranoia, catatonia, et cetera, et cetera. These things were not considered one illness as we think of them today. And that only came later under Emil Kraepelin in the early 1900s. And he unified all of these sort of concepts under the concept of dementia praecox. Now, he basically came out with this idea that there were two main psychiatric diagnoses, dementia praecox and manic depressive insanity. And he divided everything into these two groups, which we now think of as schizophrenia and bipolar disorder, respectively. Now, interestingly, he didn't divide them on the basis of different symptoms, because actually he saw, and as many of you have seen with bipolar disorder and schizophrenia, oftentimes the symptoms themselves can be very similar. People with bipolar disorder can have delusions and hallucinations, just like people with schizophrenia. And people with schizophrenia can get very, very irritable, very, very depressed, just like people with bipolar disorder. So how he distinguished them was not on the basis of their symptoms, but on the basis of their course. And he showed that people with dementia praecox, what we call schizophrenia today, had a chronic downhill course, whereas people with manic depressive insanity or bipolar disorder had a relapsing remitting course. They would have episodic illness, and between the episodes of mania or depression, sometimes were actually quite normal. So going with our schizophrenia concept, basically over the course of the next few decades, what happened is basically the concepts of Emil Kraepelin sort of collided with psychoanalytic concepts, those that came through Freud and his students and his acolytes, so to speak, and his descendants. And the concept of schizophrenia broadened a little bit to the point where we had such concepts as pseudoneurotic schizophrenia, latent schizophrenia. So we grouped many more things into the concept of schizophrenia than had previously been done. And that led to, of course, as you can imagine, a lot of confusion, people getting put on Thorazine when they probably shouldn't have been, so on and so forth. This is, during the time of DSM-2, between DSM-2 and DSM-3, let's just say the 1960s or so. And around the 1970s, that's when DSM-3 came along and really started to, there was a real sort of shift between DSM-2 and DSM-3, where it became a much more sort of codified approach to making diagnoses. And this was based on the work of people like Kurt Schneider, et cetera, and also the advent of antipsychotics, which showed that symptoms can be improved. And it was much more important at that point to have real reliability between people. When I say somebody has schizophrenia, generally speaking, someone else should as well. So we should be agreeing, and that led to a much more fine-grained approach to diagnosis, leading to what we have today, in the DSM-5. And later in the 21st century, we now start what we call deconstructing the concept of schizophrenia or psychosis. We think of it not just in terms of its, just one unitary, disease of schizophrenia with just one list of symptoms. We look at different aspects of it, like negative symptoms, positive symptoms, et cetera. Okay, so that's just, I think, a good introduction. This was published in 2009 by Rajiv Tandon. I think it's important to sort of, before diving into the technical details, what is it that we're actually talking about? What is the historical sort of like, evolution of this concept that we hold today as schizophrenia? So I don't think I need to impress upon anybody in this audience about the public health. I think this is probably, you guys probably know more about this than anybody else. The public health impact of schizophrenia is immense. It's one of the top 12 leading causes of disability worldwide. People with the illness have an increased risk of premature mortality, and the estimated average potential life loss for individuals with the illness in the United States is 28.5 years. It's often got co-occurring medical conditions such as heart disease, liver disease, diabetes, et cetera, and these contribute to the higher premature mortality rate. And interestingly, as you all know, of course, these conditions are not just comorbid, but they're possibly also caused or contributed to by the medications that we give patients. And I'll talk a lot more about this later. The suicide rate in people with schizophrenia is huge. It's about 5%, which is much more than the general population. And the cost of treatment in the United States, combined with the cost of lost productivity together, it's very difficult to estimate, but it's in the tens of billions of dollars, somewhere between 25 and $100 billion a year, which is a massive cost to this country. Okay, so let's dive into the genetics of it a little bit. So we're going to just start by looking at what we call the recurrence risk in relatives. So, sorry, let me go back here. I think I pressed the wrong button. Okay, here we go. So what you see on the left-hand side here is a classic bar graph from Gottesman. Irv Gottesman was one of the sort of founding fathers of the study of schizophrenia. And what you see here is the risk of somebody having schizophrenia if their relative has schizophrenia. And it's broken down by different relative classes. So if you see at the very top here, hopefully you all can see my pointer clearly. If you have a monozygotic or identical twin with schizophrenia, your risk is about 50%. If you're the child of two people with schizophrenia, your risk is roughly the same, about 50%. Now, if your relative, if your dizygotic or fraternal twin has schizophrenia, your risk is about 17%. If your child has schizophrenia, your risk is about 13%. If your sibling has schizophrenia, your risk is about 9 or 10%. and so on and so forth. As the genetic distance between the relatives, gets larger or increases, the risk goes down a little bit. And it goes down to the point where the baseline risk in the general population, where the prevalence is roughly 1%, maybe a little bit less than that, somewhere between 0.5% and 1%. So what does this tell us? This tells us that schizophrenia is highly familial, but does this tell us that it's genetic? Maybe somebody can answer that from the audience. We'll get just a little bit of audience participation today.
It probably, I mean, it indicates it might be, but it could be environmental too, the way they environment, the way they were raised, social factors, right?
That's exactly right, because the family members don't just share their genes, they share their environments as well. They share their diet, they share parenting, they share the susceptibility to viral disease in that particular geographic location, schooling, diet, so on and so forth. So you can't really say that it's genetic. I see somebody raise their hand.
Amy?
Yeah,
have a
so
question?
for the monozygotic twins, couldn't it be an insult that they both sustained in utero?
Well, it certainly is possible. But let's leave the individual specific case, and let's just look at the overall sort of picture where you have many thousands of monozygotic twins. What you see is a big difference between the monozygotic or the identical twins and the dizygotic and the fraternal twins, and that suggests that it is genetic.
Oh, okay.
So what you see here on the right, I'll sort of break this down a little bit. These are what we call heritability estimates. These are from different studies going back to 1941. 10 or so studies from the early 40s to the late 90s. And what these studies did is they looked at fraternal and identical twins, and from that you can actually calculate how genetic a disease is, because even though fraternal twins share 50% of their DNA, and monozygotic or identical twins share 100% of their DNA, both groups, they share the environmental sharing is about the same. So you can actually calculate how genetic something is based on the difference between the identical and fraternal twins. And that degree of something being genetic, we call that the heritability. Okay, I see a hand raised.
Yeah, can you hear me?
Yes.
Yeah, have there been any studies of monozygotic twins that were raised in different environments, so that their genetic would, if they had schizophrenia, you would attribute it to their genetic.
Solely genetics, right?
I hear you. Yes, that's a very good question. Actually, unfortunately, that's a very rare phenomenon. So there actually are very few, if any, studies that I'm aware of. There are, of course, adoption studies where somebody with the genetic predisposition to schizophrenia is adopted away to a family where there isn't such a predisposition, and they end up getting more likely to have schizophrenia like their biological family. Now, those studies have been done a lot, but not the study that you're describing so much, just because of the rarity of that particular situation. Anyway, so getting back to heritability, many studies, if you go back, these studies were sort of all over the place until about 2003. Actually, one of my colleagues, by the name of Pat Sullivan. I'm going to have to be very careful about pressing his button here. Sorry.
No worries.
Pat Sullivan, 2003, did a meta-analysis and showed that if you look at all these studies, put them all together, we can show that about 80% of the risk of schizophrenia is genetic and about 20% is environmental. Okay, so that's currently what we have. So it's about 80% genetic, 20% environmental. Overall, in some case, obviously in some cases it might be entirely, some individuals it might be entirely genetic, and other cases might be entirely environmental, but on average it's about 80% genetic, 20% environmental. But this doesn't tell us anything about the actual genes themselves. So let's move on to that. And this is kind of where things are probably going to get a little bit more technical. There's a certain type of study called the linkage study, and it's a classic type of genetic study in medicine. It was used to identify genes for cystic fibrosis and things like that. And that approach was tried with schizophrenia, bipolar disorder, et cetera, but it was not at all fruitful. So roughly in the, around 2005 or so, the field moved to something called association studies, and something called the Psychiatric Genomics Consortium was founded, also actually by the same guy who did the meta-analysis, this guy, Pat Sullivan, who sort of got these groups together. And the purpose here was to conduct mega-analyses of genetic data for psychiatric disorders. And the idea here was to put, everybody had their own little study sample, and they were all individually too small to really get much out of them as far as results go. So the only way to really make sense of all the data is to put all the data together and analyze it together. So instead of 10 people, 10 groups having small samples of 100 each, we put them all together and we might have thousands of individuals in the study, and you have statistical power that way. So this began in 2007. It initially focused on schizophrenia, bipolar disorder, major depression, autism, and ADHD, but later expanded to other disorders, including anxiety disorders, PTSD, etc. Each disorder group, so each of these disorders has their own group studying them. I belong to the schizophrenia PGC group. Each disorder group has a phenotype work group, which focuses not just on the genetics, but looks at the actual, what we call the phenotype itself, which is the clinical form of the illness. And if you look at these different groups that study these illnesses, they might use different instruments to assess symptoms. For example, we have the SCID, which is one diagnostic interview. We also have the PANS. Many of you might be familiar with these things. So our group in the phenotype work group is to sort of bring all of these, is sort of like this sort of Tower of Babel effect, where you have different groups using different diagnostic instruments. We try to bring them all together and also extract information on things like hallucinations, negative symptoms, things like that, which we can also use in genetic studies. There's also a cross disorder work group, which looks at the common genetic factors across different disorders, including, let's say, for example, bipolar disorder and schizophrenia. What commonalities do they have? So it is actually the largest biological experiment ever conducted in psychiatry, with this is 500 investigators on the slide, but it's now many more than that. Hundreds of institutions in dozens of countries, and currently we're in several millions of subjects, actually. Okay, so for schizophrenia, this work has sort of proceeded in 3 stages. PGC 1 with about 10,000 cases and 20,000 controls. PGC 2, 37,000 cases, 113,000 controls. And PGC 3, 77,000 cases and 244,000 controls. These were papers that were published sequentially over the years. Yeah, Ms. Goldstein, I see you have your hand up. I
was just going to ask about schizoaffective, which is the schizophrenia and the bipolar. How are you going to speak about how did that come to be, and what distinguishes it from bipolar with psychosis or clear schizophrenia? It's a combo, right?
Yeah, so basically, schizoaffective disorder is a diagnostic sort of category where you have the symptoms of schizophrenia and the symptoms of either major depression or bipolar disorder coexisting. It's a concept that goes back, many, many decades. It is useful in terms of, understanding the symptoms that an individual has and what kind of medications would be, useful for them. I mean, I wasn't planning on going into a lot of depth into that, but that's the general concept. And these categories can also be used in studying genetics of schizophrenia. So, for example, when we study the genetics of schizophrenia, we include people with schizoaffective disorder in the diagnostic definition as well.
Okay, thank you.
Sure. Okay, so currently we have about 90 study samples, and most of the study samples are of European ancestry, much smaller proportions of Asian ancestry, African ancestry, and Latino ancestry. But current and future work is increasingly focusing on these non-European populations, and I'll go into a little bit more detail as to why. Okay, so this is like our first technical slide here. I'll sort of walk you through this. here, but this is basically what we call a genome-wide association study. So I'll spend a little bit of time on this so that subsequent slides will make more sense. So we call it a genome-wide association study because we look at the entire genome. Now, if you look here at the bottom on the x-axis, we have the different chromosomes. So what you see here is the entire genome, all 20. We don't look at the X and the Y chromosomes, so we're only looking at 22, not 23 of the chromosomes. They're all sort of laid end to end, so to speak, schematically. And each of these dots represent a position on that chromosome that corresponds to what we call a single nucleotide polymorphism. These are thought to be sort of the spelling errors that we see in the genome. So, the bases in the genome are A, C, T, and G. We have 4 bases, and these are stringed, there are 3 billion of them in the human genome. And about every 1,000 of these bases, there's what we call a single nucleotide polymorphism, or a SNP, SNP or SNP. It's a genetic variant. It's a naturally occurring variant. It's kind of like a spelling error. So rather than an A, where an A is supposed to be, you might have a T or a C or a G. Okay. So these occur, again, in about roughly every 1,000 bases, or roughly every 1,000 letters in the genome. And these act as markers. And like I said, they're naturally occurring. And all of us have these variations, and these variants could occur. They could be very rare. They could be like 1 in 1,000, or they could be very common. They could be in 30% of us. Now, each one of these variants, or SNPs or SNPs, they have their own frequency. Some, again, are very common. Some are very rare. But what these studies do is they look at the frequency of these variants in people with schizophrenia, and they compare that with the frequency of the variant in people who don't have schizophrenia. So you can have a comparison of these frequencies. So, for example, you might have one variant that occurs in 30% of people with schizophrenia, but only 2% of people without schizophrenia. So that suggests that there is a gene close by that is causative of schizophrenia. So we mark the genome that way using these variants, and this is how we can determine the presence of a particular gene and where that gene might be. I realize that's a bit on the technical side, so if anyone has any questions about that, we can probably clear it up right now.
So do the little diamonds indicate ones that you want to pay attention to?
Yes, exactly.
Okay.
Thanks for bringing that up, because I was just about to get to that. So I'm glad I didn't forget. So I'm glad you reminded me. So basically, all of these dots here represent an individual variant in the genome, and how high it is up here on the y-axis denotes how statistically significant it is. So, for example, if you've got a variant that's 50% in people with schizophrenia and only 1% with people without schizophrenia, and you've got thousands of people in your sample, that's very highly significant. Whereas, for example, if you have something that's 33% in people with schizophrenia and 32% in people without schizophrenia, that's probably not very significant, right? So the height of these dots here is what we call the p-value. Okay, so and we're doing millions of tests here because there are millions of these variants. So you have to have a very, very significant p-value. So this is, for example, is p times 10. It's 1 times 10 to the negative 11th. This is 1 times 10 to the negative 12th, that sort of thing. So here, what we're seeing is that there are 13 variants in the genome that are what we call genome-wide significant. In other words, they're statistically significant, and they represent huge frequency differences between the cases, the people with schizophrenia, and the controls who don't have schizophrenia. So this was basically the very first study that showed that there were statistically significant genetic loci or regions in the genome. No other study had done that before. This came out in 2013. Now, this was, we had 9,000 cases and 23,000 controls. We showed 13 loci, okay? So a couple years later, just 1 year later, in 2014, as I said, this work occurs in stages as we get more and more people in our study. 2014, we have 37,000 cases, 113,000 controls. Now we have 108 instead of 13 regions of the genome. We can just say 108 genes. And most recently in 2022, we now have 77,000 cases in the study, 244,000 controls. Now we're up to almost 300 genes, okay? 278 loci or 278 genes. So this is telling us that the genetics of schizophrenia is very complex, not just one gene. Like with cystic fibrosis, if you look at, some of these really, these killer illnesses that, like cystic fibrosis, Tay-Sachs disease, things that really kill people. If you have the illness, you will probably die young, or maybe not live at all. These often occur from one gene that's very, and it gets disrupted very severely, and the person really can't live a normal life. Schizophrenia is not like that, okay? It's probably hundreds of genes, and this is very similar to what we see with many of the chronic illnesses like diabetes, high blood pressure, et cetera. These are what we call complex illnesses. They're what we call polygenic. In other words, they're caused by many, many genes. Now, we don't know how many genes there are for schizophrenia. We don't know how many risk genes there are. In this case, we found about 300, 280 or roughly 300 of them. There could be many more, but this is the current state of our understanding of the illness. We know that there are about 278 that we can sort of hang our hats on, so to speak, that are reliably assessed, and we know that they're there. Okay, Ms. Goldstein, I see your hand is up again.
Okay, I was just saying with p-values and probability, and as an engineer, we use this a lot. Could you just look at maybe even looking at the ones that are above 15 and saying these are the most probable that cause the most problems since they're the highest?
Well, that's a very good question. So to be technical about it, there's actually a threshold, a cutoff, and that is 1 × 10 to the -8th.
Okay.
So 0.00000001, something like that. And that is what we call the threshold for genome-wide significance. Because we've done millions of tests, you can't just, the ordinary p-value statistics, as everybody knows, is 0.05. But that's if you do one test. Now, if you do a million tests, you have to have a much more stringent threshold. So you have to have 1 × 10 to the -8th, which is like 8. You were saying 15, but it's really 8 that's the cutoff. Now, you're going to see a lot of these that are much more significant than that. So we have this gene right here that's significant to, it's basically .3901, so to speak, okay? So that's a very good question. But to get technical about it, it's actually the 8 here, or the p-value of 1 × 10 to the -8th, that's considered significant.
Where the line's drawn.
Exactly. Exactly. That's where this one.
Yeah, we had a couple questions in chat. One was, do you think this will lead to genetic therapy for mental illnesses? That's one. And what is the correlation of schizophrenia and viral pneumonia in the 9th month of pregnancy or other trauma surrounding childbirth? Two very different questions.
These are very different questions, and I realize, I'm probably, harping on this a little bit because I want to move on to more, clinically relevant points, which I will get to. I just wanted to make sure people sort of understood, what the sort of basis of all this is and how we're reaching these conclusions. So to answer the first question, do I think that there's going to be gene therapy in the future for schizophrenia? I know that there is going to be gene therapy for many things, including a lot of neurological illnesses. Now, will it exist for schizophrenia? I think it's quite likely, but I can't say that for sure, simply because, as I said, we're not looking at one gene. We're looking at hundreds of genes. So if you want to do gene therapy, should I give an individual, should I inject them with 300 genes using a viral, using whatever vehicle it's used? I'm not so sure. Now, it might be that this, hypothetically, it might be that this gene right here that's very, very highly significant, maybe that one, if I do gene therapy for that gene, maybe that will be effective. That's a very good question, and I wish I could, I think it's all speculation at this point. I would like to think so at some point. So the second question, if I understand it correctly, I wasn't quite sure what the actual question is, but related to the viral hypothesis, intrauterine viral infection.
Yes, if it was related to 9-month viral pneumonia in the 9th month of pregnancy. That time, isn't everything developed? I would think it would be earlier if it was going to be impactful.
Right. Well, it's the second trimester that is considered to be the sort of risk period. So the viral hypothesis is, one of many hypotheses of schizophrenia. And there certainly is known to be an excess of winter births. So there's more people with schizophrenia are born in winter than would be expected by chance. So that's the sort of the basis of the hypothesis. And it's the second trimester that's actually considered the risk period.
Thank you.
Sure. Okay, so again, this is kind of where we're at right now. We have 280 genes. This is how we know that these genes are the ones in question, and I kind of wanted to illustrate how we know these things, and that's kind of where we're at. So let's move on to the next one. So next slide. So what are the primary findings? As I said, and I'll probably breeze through some of these other slides because I think probably for this audience, you guys might have more of sort of a clinical, more real world practical applications in mind. Again, 287 loci were identified. Separate analyses for men and women resulted in a genetic correlation of 1. Now that was actually an interesting finding. Now, one question that we had before was, are the genetics, whatever genes there are they different in men than women? No, that's what this study showed. They're about the same. And we showed that using all of the genetic information, we actually only showed a heritability of about 24%. Now, you might remember in my early slides, I showed that the twin studies showed that the heritability was 80%. These studies showed that it was 24%. And the reason we think that's the case is because we're only looking at we're only scratching the surface of the genome. We're only looking at a very small proportion of genetic changes. We're not looking at very rare events. We're not looking at what we call copy number variants and things like that. So we're only scratching the surface of the genome. And our understanding of how genes work together is also very, very incomplete at this point. They're not what we call univariate. It's not just like one gene by itself. They interact with each other. And we haven't really gotten to the point where we understand that interaction yet. And we haven't been able to model it. In any case, going back to the results, we also showed that it was the individuals who were more severely affected that tended to have the higher genetic risk. So people who had been hospitalized, people who had been treated with clozapine, et cetera, et cetera. I just want to go back to make one more introduce one last concept moving forward, and that's the concept of the polygenic risk score. It's actually a very interesting concept that has been sort of developed over the last 15, 20 years or so. As you can see here, there's 278 loci that we have identified, right? And at each of these loci, there's a spelling error, right? Mm-hmm. And so an individual can have either you can have 0 or 278 of these mm-hmm. spelling errors, right? You can actually count how many of these spelling errors of these 278 loci an individual has. I might have, 100. Somebody else might have 130, right? So this introduces a way where we can now quantify the degree of genetic risk an individual has. And it's been used in some very creative ways now. We can actually look at the relationship between different disorders, et cetera, et cetera. So the concept is the polygenic risk score, or the PRS. And again, that's defined as how many of these spelling errors an individual has. So if there's 278 loci, in theory, you can have anywhere between 0 and 278 of them, an individual can. And that is considered your PRS, your polygenic risk score. Okay, so what we're, again, we're gonna get a little bit technical here, but I'll try to, make it, more practical. What genes were involved, and the genes that seem to be involved were involved in the development of the body and the brain, the differentiation of different tissues from each other, and importantly, in the synapse, the synapse, which is the space between the two neurons, how two neurons communicate with each other, as you all know. There's a space between them called the synapse, and how they communicate is through neurotransmitters. This neuron, the neuron that communicates is the one that sends out neurotransmitters to the neuron that is communicated to, right? And so that space between them is called the synapse. And what we found is many of the genes of these 278 genes that were found were actually genes that either hold the synapse together or somehow are involved in synaptic transmission. That basically ended up being where a lot of the convergence was of where these genes are. So if you look at different disorders, Parkinson's disease, et cetera, Alzheimer's, Huntington's disease, they involve certain regions of the brain. For example, Parkinson's disease, it involves a certain region of the brain called the substantia nigra, where you get death of certain cells. That's not what we're seeing in schizophrenia. We're not seeing a particular part of the brain that has a lesion or that has like, an area of dysfunction. We're actually seeing the dysfunction at a very, very microscopic level. It's where the neurons communicate with each other. That's kind of where it's not a macroscopic lesion as we see in neurological disorders. It's a very microscopic disorder, if that makes any sense. Yes.
It's interesting because I've heard at psychiatric conferences that there's thoughts that it is a timing issue with schizophrenia. So I'm wondering, when people think they're hearing voices and whether they're outside voices or inside voices, could it be that lack of quick synapses delivery that adds? I wonder how many people with executive functioning problems also fall into the category of potentially developing schizophrenia.
Well, sure. So, so when you look at dysfunction, I mean, it can take place at many different levels. At a very microscopic level, you're seeing, some kind of dysfunction in the way that neurons are communicating with each other. Now, that might manifest itself at a circuit level. If you look at, at the level of, different parts of the brain communicating with each other, that will manifest itself in some probably unpredictable ways. So, the way I see it is you might have a dysfunction at a very microscopic level that somehow has ramifications at a much larger level. And I think that's maybe that's what you're getting at. Yes, the executive function issues probably do come about at a certain point at a dysfunction at the synapse. Something at the synapse is not working correctly, and that ends up leading to dysfunction at a sort of like a brain network circuit level, and that leads to what we see as executive or executive dysfunction or problems with executive functioning, if that helps.
Thank you.
Okay. All right, moving on. So as I was saying earlier, the vast majority of previous genetic studies have been done in European ancestry populations, and we're now trying to move away from that to sort of get more, ethnic groups involved, including African ancestry and Latino ancestry and Asian ancestry populations. And we're actually doing some of that work right here in Phoenix. So we're working on a study called the Genomic Psychiatry Cohort. I'm the principal investigator here in Phoenix, but it's a multi-center study. We have sites in Utah, Rutgers, in New Jersey, and a few others around the country. And actually, many others have contributed over the years. So currently, it represents the largest collection of African ancestry or African American schizophrenia and control subjects that has been studied to date. We currently have about 6,000 schizophrenia cases and 4,000 controls. We're also doing this study in Latino ancestry populations. And I'll be honest with you, one of the reasons that I came to Phoenix 3 years ago from New York City was the opportunity to sort of branch out into these other populations. Of course, you know, there's a very large Latino population in the Southwest, certainly in Arizona. And this, to me, represented a real scientific opportunity to branch out. And it's one of the reasons that convinced me to come here 3 years ago. Not the only reason, but one of them. So it is very important to what we're doing here at the University of Arizona to really try to you know, take our genetic studies to one of the next levels, which is to examine other ancestry groups. And I'll show you why that's important in a minute. So, but what we've seen so far, and I won't go through a lot of detail here, is that there's a lot of consistency of effect across the different ethnic groups. So, for example, you know, let's go back to the SNPs or the spelling errors. If a spelling error is a risk, it's a risk factor in one population, it's probably a risk factor in another population. We're not seeing opposite effects in different ethnic groups. However, we are able to see different new genes are popping up when you look at different ethnic groups that we weren't able to see in European ancestry, for example. And there's another reason that's interesting here. Let's ignore the left-hand side of this slide. Let's look here on the right. I know it's kind of, you know, kind of abstract looking, but let's look at this. Can you guys see my pointer?
Yes.
Okay, so what we're seeing here is if you look at those plots I showed earlier and zoom way into a very small region. This is a very small region of the genome. There's only a handful of genes here. What we're seeing is, again, the statistical significance of these dots. There's the line of genome-wide significance. But what we're showing here is, when you look at in the pink, we're looking at just one ethnic group, and in the red, we are looking at all of the ethnic groups combined. So what you're seeing here is our confidence interval, or where we think the actual gene is. We can narrow that confidence interval by adding more and more, you know, additional ethnic groups. Mm-hmm. And so what we see here is a narrowing. If you look at the pink, it's a larger band than the red. And down here, if you look at this area, this is another region of the genome where we actually were able to narrow down from looking at just the European ancestry group by itself in pink down to the combined group in the red. Now, we narrowed it down to one very, very small region of the genome. So the take-home point here is, when you look at just one ancestry, you're actually gonna have much less precision in terms of where you understand the gene to be. Okay, so your confidence interval can shrink dramatically to a much smaller area if you look at more additional ethnic groups. Because what you do there is you introduce a lot of heterogeneity, a lot of variation, and that actually increases your precision. Okay, so it's been a real advancement for us to introduce other ethnic groups into analysis. It's not just a matter of, it's not a DEI issue only. It is really a very scientific issue in terms of being able to you know, identify our genes with much greater precision. Okay.
Wouldn't it also help cancel out some of the inbreeding of some of the European societies if you're looking at other genes and seeing commonality? You can say that these other mistakes, though they're there, may not be as significant.
I think that you're getting at pretty much the same thing that I was saying. Yeah. Thank you. Okay, so I'm going to skip a little bit because I realize I've probably got a lot more slides here than we have time for. So
I think we're all very engaged, so it's up to you how long you want to go. But to me, it's fascinating to actually see actual research going on in this area, which I think there's so little that has been done.
Please proceed. It's very interesting.
Thank you. Well, I'll just skip this part, which is about using sequencing. It's I'll get to some more clinically relevant area. I guess you guys probably don't know how many slides I have. Why are you saying that? I have tons of slides here, but so I'll just skip this area, which talks about gene sequencing as opposed to the genome-wide association study, which it allows us to look at much more rare spelling errors, so to speak, like ones that exist, like 1 in 10,000, so on and so forth. But it's basically telling us more or less the same thing. So I'm not going to dwell on it too much. So let's go on to this here. So, so one question that we've had is, can genetics help explain phenotypic complexity? Okay, so what is phenotypic complexity? Again, the phenotype, as we were talking earlier, is not, it's not the genes. It's actually what you can see yourself. Pheno, like phenol, which is like a, you know, something that lights up. Epiphany. These are concepts that stem from the Greek word. I think it's phen, which refers to that which is visible. Okay. So the phenotype is what you actually see. So whether the person has schizophrenia or not, whether they have black hair, blonde hair, whether they have blue or brown eyes, whether they have male pattern balding or not, that's what we call the phenotype. So in psychiatry, we have something that I call phenotypic complexity. So as I said earlier, and as many of you as clinicians have seen, you know, people with bipolar disorder and schizophrenia will often have the same exact symptoms, just not at the same time, right? People with major depression can have delusions and hallucinations, okay? So we're seeing a lot of complexity in terms of what we actually identify as the illness. So in addition to that, we're seeing within the same illness, you can have lots of different types of presentations. So like you can have similar symptoms across different illnesses, but within the illness, you can also have many different types of presentations. You can see people with schizophrenia who have high levels of negative symptoms. You know, negative symptoms, as many of you know, things like a lack of self-care, lack of interest in social interaction. These are the things that prevent people from having jobs and having, you know, fulfilling relationships, getting married, things like that. We also have hallucinations, we have delusions, and in any particular patient, you can have any combination of these things. You can have patients that have nothing but hallucinations and delusions, and you can have patients that have very little in the way of hallucinations and delusions, but lots of negative symptoms, right? You can have patients who have lots of disorganization, disorganized thought, disorganized speech, you know, people that you can't understand because the grammar of their speech has been completely degraded to the point where it's word salad. I mean, I'm sure all of you can relate to what I'm saying. Anyone who's seen people with serious mental illness you know exactly what I'm talking about. So my mentor, Ken Kendler, back in 2005, this is back when I was a fellow, we came up with this idea that there are different types of ways that genes can act. We have a susceptibility gene, which increases the risk of the illness, but it doesn't change how an illness presents itself. So, you know, it increases your risk for schizophrenia. It's a risk gene for schizophrenia, but it doesn't affect, you know, what kind of symptoms you see in the person. There's also what we call the modifier gene, which affects the symptoms themselves, but it doesn't actually affect your risk of the illness. So let's just say you already have the illness. A certain set of genes might come online, which affect your levels of negative symptoms, your levels of hallucinations, levels of mood, manic and depressive symptoms, so on and so forth. We call that the modifier gene, okay? And then we call, we have a category called the susceptibility modifier gene, which is a gene, it's a risk gene for the illness, but it doesn't just give you the illness, it gives you a particular type or a particular presentation of the illness. For example, high levels of negative symptoms or low levels of negative symptoms and high levels of hallucinations. Okay, and then we have finally the overlap gene, which is a gene that can increase the risk of more than one illness. It can either lead to bipolar disorder or schizophrenia, depending on what other factors might be present. Is this kind of a clear present? Is this clear to everybody, or I know this can be a little confusing?
I think it's great, but I love bar graphs and science. So
okay, great. So these are the concepts. We have the susceptibility gene, the modifier gene, and the susceptibility modifier gene, as well as the overlap gene. So what we've seen here in this bar graph right here, we've looked at as I said earlier, the heritability you know, how genetic a particular disorder or condition is. And these are based on not on the twins, but on the genetic data. So for schizophrenia, as I said earlier, it's about, you know, 25%. And these are the different heritabilities of the different disorders based on the actual genetic marker studies. So for ADHD, it's about 27%. Bipolar disorder, it's about 25%. Schizophrenia, 24%. They're all of these disorders have heritabilities, but we can also calculate their co-heritability. Now, the co-heritability is defined as the degree to which the two disorders share their genetic risk factors. Okay, so for schizophrenia and bipolar disorder, as a pair, let's say, their co-heritability is about 15%. So that tells us basically that the genes that cause schizophrenia and bipolar disorder, about 15% of them are jointly risk genes or in common to the two disorders. Okay,
So you're really unlucky if you get that one, because you have you can have all the schizophrenia and all the bipolar and all that symptomatology of each disease.
Basically, right, exactly. So basically what this is saying is that 15% of the genes for schizophrenia and bipolar disorder are actually shared between the two disorders. And you can see the same thing for many of the other, excuse me. I thought I had gotten the hang of this already.
No worries.
There we go. Okay. So the point that this slide is trying to make, and this was actually a very impactful paper that was published in 2013, is that many disordered dyads are co-heritable. Okay. So in other words, there's a lot of overlap genes. There's not just one set of genes for schizophrenia, one set of genes for bipolar disorder, one set of genes for major depression. Chances are there's a whole set of genes that are actually in common across the psychiatric illnesses, and they increase your overall risk to psychiatric illness. And there might be another set of factors, either another set of genes or another set of environmental factors, which sort of determines which psychiatric illness you end up getting, you know, either bipolar disorder, major depression, or schizophrenia. Okay, so that was actually a very interesting finding that we had about a decade ago or so. Okay, so now, again, as I said earlier, within illness, you also see a lot of variation. So what we did, this is a study that was done by our group. It's currently it's been submitted, has not been published yet. What we did here is we took a large group of people with schizophrenia and clustered them into different subtypes based on their symptoms, not based on the DSM subtypes, like, you know, paranoid schizophrenia, disorganized schizophrenia, et cetera. We actually took their symptoms and we did what we call a cluster analysis, and we came up with 4 clusters. Okay, so just looking here, cluster 1, we called it the severe cluster. Okay, so 0 is the mean, and green is negative symptoms, purple positive symptoms, orange is mood symptoms, yellow disorganized symptoms, and blue is excitement or manic symptoms. And that cluster, they had higher than average for everything. So we called them the severe cluster. Okay, cluster 2 was basically a high negative symptom cluster. Cluster 3 was a mild cluster. Everybody scored below the mean on everything. And then cluster 4 was a low negative symptom cluster. In other words, they scored high on everything except for negative symptoms. So we basically divided the people with schizophrenia into these 4 clusters, and then we tried to see if there were genetic differences. And what we found here is that cluster 2, the low negative symptom group, actually had a lower polygenic risk. So remember, I introduced that concept of the polygenic risk score. Everybody has sort of a score. If you look at somebody's genome, if you look at my genome, look at your genome, we can quantify, how many of those spelling errors you have, because even those of us who don't have the illness, we also have those same spelling errors. We just have fewer of them. Okay, so what this study showed us is that there are certain subtypes of schizophrenia that actually differ not only clinically, but also genetically, okay? So this is the first time, that we showed that you can actually show that certain individuals with certain presentations actually have slightly different genetic signatures, okay? So that, again, goes to the heterogeneity. The heterogeneity of the illness is not just seen at the symptom level, but it's also seen at the genetic level. All right, so moving on to the, precision medicine. Back in 2015, President Obama came out with the Precision Medicine Initiative, and I'll just quote him right here. And he's basically, when he inaugurated this, and I quote, "Doctors have always recognized that every patient is unique. Doctors have always tried to tailor their treatments as best they can to individuals. You can match a blood transfusion to a blood type. That was an important discovery. What if matching a cancer cure to our genetic code was just as easy, just as standard? What if figuring out the right dose of medicine was as simple as taking our temperature?" So the U.S. government now has spent about a couple $100 million on the Precision Medicine Initiative. So I wanted to talk about the precision medicine ecosystem. First of all, sorry, 1 second. Let me get my slides together again. I apologize. So precision medicine is basically an approach to medicine where the practitioner or the healthcare system can look at your genes, your DNA, and all of the other factors, your diet, whatever factors we can actually identify about an individual to get a more complete picture of them, both genetically and environmentally. Can we use that information to identify what's the best treatment for them? That's the whole idea behind precision medicine. So this is sort of like a schematic of what we call the precision medicine ecosystem. I mean, I bet everybody, including myself, we've looked at your patient portal at some point. So the patient can access their patient portal, which leads to their electronic medical record or electronic health record, EHR. The EHR can lead to a case level database, and researchers can access that. Other institutions could potentially access that as well. The individual, the patient, also gives blood for a biobank, a patient sample, and that is banked. And through lots and lots of research, we can identify relationships between drug response and genes and things like that. And eventually, all of that goes to a curated knowledge base, and that can be displayed at the point of care. Your clinician can see that when they're seeing you in the office, and based on the genes that you have, based on other factors, your diet, so on and so forth, all of that information can be put together to identify what are the most likely medications that can help you. For example, would it be Prozac or Wellbutrin? Which one is more likely to cause you side effects? Which one is more likely to be effective? That's the whole idea behind precision medicine, and that's what we call the precision medicine ecosystem. So this came out in 2015, sort of a schematic, but it's been updated more recently. So this is what we're hoping to look at in 2030. The precision medicine ecosystem will also be benefited by big data and artificial intelligence. There will be routine clinical genomics. The electronic health record will be readily available. We're also going to be able to access what we call phenomics in the environment. We'll be able to access people's heart rates and things like that at home while they're living in the real world, not just in the office. There's going to be a lot more, hopefully, a lot more attention paid to privacy, trust, and return of value. And we're going to have huge longitudinal cohorts and a lot more ethnic groups involved and more diversity and inclusion. So this is kind of what sort of is envisioned to be the precision medicine ecosystem, not just today, but also in the future. Okay, so moving on to something of much more practical importance to all of us, treatment resistance. And we're gonna see now how current precision medicine modalities are being applied to something very, very near and dear to everyone's heart here, including my own and all of you, I'm sure, treatment resistance and schizophrenia. As many of you know, there's a huge number of patients that don't get better with no matter what you give them. And I worked at the DCVA Medical Center for 15 years in their depot neuroleptic clinic, working with chronic schizophrenia patients, and I've seen this with many hundreds of patients. So we know that 20 to 30% of people with schizophrenia don't respond to treatment. And the only widely used therapy for treatment-resistant schizophrenia right now is clozapine. However, only 60% of patients respond to clozapine, and the delay in getting patients on clozapine and getting them prescribed can be associated with even more treatment resistance. Now, the biological basis of treatment resistance is unclear. It's possible that the PRS, the polygenic risk score, in people with treatment-resistant schizophrenia might just be, extraordinarily high. That's possible. It might be that clozapine's efficacy is related to some underlying biology of treatment resistance that we don't quite understand. However, the genetic studies of treatment resistance have really not been done yet. We're only just getting started. So moving on to the next slide, I wanted to show you one of the very first studies of the genetics of treatment resistance. And this is kind of where genetics and precision medicine sort of come together. So what you see here is another Manhattan plot. We call this the Manhattan plot. You've seen a bunch of these already, right? Remember the line, the dots, and the chromosomes, and all that here. But what we're looking at here is not a genetic study of schizophrenia. We're looking at a genetic study of treatment resistance. Okay, so instead of cases and controls, people with schizophrenia, people without schizophrenia, what we're looking at here, we're comparing people who respond to people who don't respond to treatment. And what we're trying to identify are genes that have something to do with treatment resistance. And Ms. Goldstein, as you pointed out, there was that red line. Now you don't see a red line here, right? So in other words, we did not find any specific gene that that was let's just say a gene for treatment resistance, or a genetic risk factor for treatment resistance. However, let's go back to our old friend, the polygenic risk score. If you create a polygenic risk score based on this information, even though you don't have a single gene that is significant, what we're actually seeing is that you can actually predict treatment resistance in a totally independent sample. And that's what these bar graphs right here are showing you. Actually, these graphs right here. So using this information, we were able to predict treatment resistance in these two samples. However, we were not able to predict schizophrenia risk in this sample. So that suggests that whatever the biology of treatment resistance is, it's probably different than the biology of schizophrenia. So again, this is the very first genetic study of treatment resistance. It does show that there is actually a genetic signature of treatment resistance, even though we have yet to identify an actual single gene. We have identified probably a bunch of genes in here. We just don't know which ones they are yet because our sample size isn't large enough yet. So I hope that wasn't too confusing, but yeah, Ms. Goldstein, you have a question. I'm sorry, I can't hear you. Are you muted?
Yes, I was. One thing that I was thinking about, maybe if you didn't find it, are you looking at the age of the people in the study with schizophrenia? So maybe first episode people, young people, versus ones that have been in psychosis for years and maybe they've developed or they're more chronic. I wonder if that's the independent variable that may be confusing here.
That's a very good question. So it sounds like your question is maybe we're not looking at the right treatment resistant cases.
Or you're not looking at another variable. If you
Right.
add that variable in and rerun just the data, maybe it would plot out differently.
That's a very good question. Now that's certainly a possibility. However, what we can say is that we also, if you go back to those earlier slides, we had 27,000 cases, 100,000 controls. We're not anywhere near that for treatment resistance because now we're looking at people with schizophrenia and we're looking at their drug response, which is a much more expensive analysis, to be honest with you, because you have to follow them over time, see whether they respond. It's a very different analysis than looking at whether they have schizophrenia or not. So our numbers here are much smaller. So we can't say that we have the statistical power yet But what we can say based on this, based on the fact that our polygenic risk score is actually predictive in an independent sample, is that we are onto something. We are seeing the effects of the genes, but not individually. We're seeing the effects of the genes all together in tandem.
Yeah.
So
Globally.
you are probably right to an extent, but we're also not yet at the point where we have statistical power. And we're just at the very beginning of our studies of treatment resistance. This is the very first one.
This is fascinating. Amy, you have a question?
Yeah, more comment because, you know, our treatments up to now have been pretty homogeneous, you know, very few different mechanisms of action with the medications. So it's really just treatment resistance to what we currently have.
Correct. You're absolutely right. So, you know, chances are there are molecules out there that we just haven't discovered yet. You know, there are clozapines that are much better than clozapine, and that don't cause illnesses. And that's where, you know, drug discovery is an extremely important endeavor. And it's, you know, and rightly so, you know, it's, you know, billions of dollars have been spent on it. I think you have a good point as well. So hopefully we will identify those molecules that are much more effective. And, you know, we are coming out with more and more molecules, you know, as time goes on. Some recent ones have been identified and they're being tested and marketed, hopefully marketed more. Okay, so the take home point here is that we've now been able to use our genetic tools, not only the, you know, genome-wide association studies, but these analytic concepts, the polygenic risk score, et cetera, to now identify genetic signatures for treatment resistance. So it's we're at the very beginning of this, but we are confident that we are onto something. And eventually, hopefully, we'll be able to use this kind of information to sort of predict who is treatment resistant before we, you know, treat them for 10 years and find out that they're treatment resistant. You know what I mean? We'll be able to identify these people earlier on and institute preventive measures, maybe move to clozapine quicker in certain people, so on and so forth. Okay. So I'm going to go on to the Million Veteran Program, which is my predominant study sample right now. The Million Veteran Program is funded by the VA, the Department of Veterans Affairs. I'm a VA investigator myself. Even though I'm chair of the department at the University of Arizona, I also work for Banner. I also work for, so I serve 3 masters. I've been in the VA system the majority of my career. My father was a veteran. He was an Air Force veteran and got his treatment at the VA, actually for bipolar disorder. So I've always sort of felt sort of close to the veterans in the VA system. I've worked for the VA system for many years. But in addition, the VA has one of the richest research data sets in the world, and I'll tell you why. So the Million Veteran Program aims to create a longitudinal cohort of 2 million veterans at about 100 VA sites. We have 650,000 of them genotyped to date, meaning we have their genetic data, not just their blood samples. The participants donate blood, they consent to future contact and electronic medical record access. They complete surveys on lifestyle, military exposure, et cetera. And they're genotyped using a customized Affymetrix Axiom Biobank array. That's just the technology that's used to identify each individual spelling error, so to speak, okay? So in addition to that very rich genetic data set, we also have in the VA system one of the world's finest electronic medical record systems, and it's been around longer than any other in the United States. And I just kind of wanted to show you, this is a screenshot of one of my own patient that I treated at the Washington, D.C. VA Medical Center for about 13 years. And what you're seeing here is his weight over time. Can you all see the numbers here? Are they big enough to read?
He's really heavy.
Well, he didn't start out really heavy, as you can see, right? I mean, his very first reading in 95 was at 135, and progressively he gained weight to the point where he got to be 285 pounds. I mean, that's just an incredible amount of weight gain, and more than any. This is okay. Give you a little bit of background on this guy. The worst probably the worst case of schizophrenia I've ever seen. He was a U.S. Army veteran. He served in the Army. He was actually quite highly educated. His mother was French, so he's bilingual in French and in English. He studied art in Paris, and he was a guitar player, actually a pretty good guitar player. Went to the military, became schizophrenic in the military. I'm not sure exactly what happened to him because he couldn't really communicate very well. But he basically developed a very high negative symptom variant of schizophrenia where all he did all day was sit in his room and stare at the wall, essentially. No interest in human interaction, no interest in taking a bath, no interest in really doing anything for himself. Probably the most deteriorated case that I've ever worked with outside of the state system. His mother and his sister basically took care of him. They brought him food. They, convinced him to take a bath every few weeks, so on and so forth. And a very, very, very deteriorated psychiatric case of schizophrenia. In addition to that, the worst case of antipsychotic induced weight gain I've ever seen. So as you can see here, his weight spiked from somewhere in the hundreds to 285. And you can see there are a lot more readings here. This is about the time where I actually put him on a new medication, Consta, risperidone Consta, because this was back in the early 2000s, and I was this freshly minted psychiatrist out of NYU, and Consta had just came out, and it was exciting to get everybody on this this second generation LAI antipsychotic, and everybody will get better. But this particular poor guy did not really get much better, but his weight skyrocketed. And in addition to that, his cholesterol, I'm not showing the cholesterol slide, but his cholesterol spiked to 800, if you can believe that. And I got it tested again, and it was still 800. And I thought I was going to have a heart attack myself. So point I'm trying to make is this can be a very, very severe side effect. It shortens people's lives, and I'm sure many of you have seen it in many patients. Antipsychotic-induced weight gain, it convinces some patients to get off their medications and never come back, shortens lives. It's terrible. I mean, I don't need to convince you of that. So what do we do about it? So what we did, first of all, going back again to the whole idea of precision medicine and where sort of genetics collides with clinical psychiatry, what we did here is we actually did a genetic study of antipsychotic-induced weight gain. So what I'm going to show you here is, these are my last couple of slides. This is unpublished data. I just presented this data in Singapore about a month ago at the World Congress of Psychiatric Genetics. I also presented it in Florence in April at the Schizophrenia International Research Society meeting. We're in process of finalizing this work. So these are not as pretty pictures because they have not been fully fully completed yet. But again, these are Manhattan plots, and what we're looking at here, we're not looking at schizophrenia versus control, and we're not looking at treatment resistant versus treatment responsive. What we're looking at here is people who gained weight versus people who didn't. Okay. And you don't see this is again the famous red line that Ms. Goldstein pointed out. We're seeing 4 genes at that red line in people with European ancestry, 3,000 cases, 21,000 controls. We're seeing a couple of genes also with people of Hispanic ancestry, 392 cases, 3,000 controls. Now these genes are different in these two populations, with the exception of this one right here on chromosome 1. And so we had not seen this kind of signal before. So we went down, drilled down into this region of chromosome 1, and we showed that it was basically the same gene in these two populations, Europeans up here and Hispanics, Latinos down here. And it's the same exact gene. I'd never heard of this gene before. It's called PDRM16. So we looked it up, and what we found is that this gene determines the cell fate of brown adipocytes. You guys probably know the distinction between brown fat and white fat. So basically, this gene determines stem cells to become brown fat as opposed to white fat cells. It was the first such study that identified this gene or even suggested this gene might be important in antipsychotic induced weight gain. And so this is kind of where we're at right now. We are at the point where we're able to use this genetic information to look not just at the risk factors for schizophrenia, but we're also looking at risk factors for treatment resistance and antipsychotic induced weight gain. And the hope is that we'll be able to use this kind of genetic information to identify people who are at high risk of these things and maybe put them on different medications. We're at the very early stages of precision medicine in psychiatry and schizophrenia, but as you can see, we are really at the beginning of a very exciting era where we're able to actually make significant findings and hopefully expand on these findings to help patients in terms of identifying good treatments for them, ones that are less likely to cause them side effects.
So back on the slide before, was that African, the third that didn't have the weight gain?
Well, they had the weight gain. They had the weight gain, but we just did not identify. We didn't
above the red. Yeah.
Didn't get anything above the red. They had the same degree of weight gain. We just weren't able to identify the genes.
Okay.
Good pickup, by the way. And I'm not sure why that's the case. We're going to have to go back to that and look into it more thoroughly. I mean, again, my intention in this talk was to give you a mix of old data and brand new fresh data as well. So this is very fresh data, and we haven't completely figured out what's going on here, but we have identified this one gene which had never been identified before that's not involved in feeding behavior. It's actually involved in the development of the fat cells themselves. So the take-home point for me as a clinician is that the underlying biology of antipsychotic induced weight gain may involve not just feeding behavior, but it might involve the actual, biology of the fat tissue itself in certain individuals. And so, I'm going to go ahead and skip this slide. So we're sort of at the end. I'm just going to give you one last slide as a teaser. As I've mentioned, I've made sort of I've alluded to, or I've made reference to the electronic medical record over and over again during this talk. And one of the things that we are interested in doing is actually being able to extract clinical information from the electronic medical record. So the idea here is, can you extract meaningful clinical information from text notes in an automated way, not just through reading the record? So for example, as a psychiatrist, I get a patient come to me, I read their electronic medical record. It might take me an hour to go through all of the doctor's notes and to try to figure out what this patient has, what their previous treatment was, what symptoms they've had, et cetera. And if I'm doing research, I'll have to extract that information by reading it, checking off boxes and say, well, this patient has this particular profile, let's put them in this category. Now, can I do that now that we have AI and much more powerful tools? Can we actually use some of these tools to extract this meaningful information in a much quicker way? Because it might take me an hour to read a patient's, a thick chart. However, it could take a computer, fractions of a second, and maybe a computer could do it on the scale of, hundreds of thousands of cases, and do a much more efficient job. So what we tried to do here is we tried to see if our current large language models, including ChatGPT, can they be used to extract clinically meaningful information? So what we did is we took 24 case histories from the DSM, this book right here that I have on my bookshelf, the DSM-5 case book. And I had one of my trainees go in there and rate each case for the presence of these symptoms: non-affective hallucinations, grandiose delusions, persecutory delusions, loss of pleasure, dysphoria, irritable mood, restricted affect, excessive activity, and bizarre behavior. And we had her rate each of these cases on the presence of these symptoms, yes, no. And then we had the large language models, in this case, Llama 2, which is the Facebook algorithm, ChatGPT 3.5, and ChatGPT GPT-4 do the same thing using the prompts for the definitions of these items. And we tried to see what the concordance was between each of these large language models and the human. So what we saw is in blue, ChatGPT-4 did a pretty good job, actually. Almost too good of a job, to be honest with you. In this case, persecutory delusions had a 100% concordance with a human rater. ChatGPT 3.5, on the other hand, did not do a very good job. On average, roughly 50% concordance. The Llama 2 did a little bit better than ChatGPT 3.5, not quite as good as GPT-4. So what we're seeing here, if you look at ChatGPT-4, on average, there was about 75% concordance with the human rater for these symptoms. And, these large language models are just getting better and better. So the take-home point here is that we're at a point in the technology where we can combine genetics and AI to now do much more powerful genetic studies than we were ever able to do in the past. And it's really just it's a brave new world, in some ways scary, but in other ways, it's opening doors that we had not been able to open before in terms of doing the research. So I'll go ahead and stop here. And our conclusions, I'll just a couple of brief conclusions here. So schizophrenia, like many non-psychiatric disorders, is polygenic, and the risk is conferred by both common and rare variants. The clinical heterogeneity is due in part to genetic heterogeneity. As I mentioned earlier, there are modifier and susceptibility genes, et cetera. It shares genetic risk variants, both common and rare, with other psychiatric disorders, as well as a number of somatic illnesses. The results from European populations can't be fully generalized to non-European populations, and more studies of non-European ancestry are needed. And we're now able to identify genetic signatures of drug response, both beneficial and adverse effects, using genomic and large-scale electronic medical record data. And hopefully this information can guide the development of precision psychiatry modalities, maximize benefit and minimize risk to patients. And so that's it for me. Those are all my slides, and we can stop here. Thank you for your time, and certainly happy to spend some time answering questions. Ms. Goldstein, I see you have your hand up.
Yeah, my question was on, there's a big difference too between the different models of GPT-3, 3.5, 4, but they also have enterprise models that corporations and universities are using, and those are even much more accurate. So do you think Banner will get an enterprise level AI system that could be really finely tuned to help with some of this research and note taking, and also kind of fill the gap where they're using nurse practitioners that don't have the same training as psychiatrists to even help them in their clinical practices?
Yeah, no, it's a great question. I'll be honest with you, we're not at the point yet of applying these methods to the Banner data. I mean, this is all VA data.
Okay.
I believe the answer to your question is yes, eventually. So just to let you know a little bit about this particular study, we actually did this on the Oak Ridge National Laboratory supercomputer, which is the fastest computer in the country, in the world, as a matter of fact. The VA has put all of its entire 25 million veteran EMR data set onto that computer. And that's the only place where we're allowed to actually analyze this data. And they are not actually currently allowing us to use sort of these enterprise large language models. So we're actually really limited to the Facebook algorithm at this point, which is called Llama 2. So we're currently in a state where we're working, with a number of sort of limitations that we have on us from not just from Banner, but also from the U.S. government. We can't use ChatGPT on this data set yet. So you have a very good point. These models are getting better and better. And I believe, yes, we will get to the best of the models eventually, where we're just not there yet.
I think fascinating talk to, we did have some comments. Again, I think some of them that have been entered.
I'll go ahead and
I'm
stop.
looking through to see if there's any anything. Oh, that one was about it takes an average of 11 years to get a diagnosis? Is it because symptoms overlap at times? Your thoughts on that?
Sure. I mean, in my experience, early in the course of schizophrenia, you get a lot more sort of, less of the, again, if you go back to Kreplin, he differentiated schizophrenia and bipolar disorder on the basis of their course. And sometimes you have to see that sort of relapsing remitting course in bipolar disorder or a chronic downhill course in schizophrenia. And earlier on, you get more mood symptoms in schizophrenia, and later you get more and more negative symptoms. So I do think some patients, if their clinical presentation evolves over time, and you need to watch for that pattern, and it just takes a while for that pattern to emerge.
Any other questions for Dr. Fanous? Anne?
Hi. I'm very impressed with the amount of research that has occurred, and I'm wanted to ask you if there's any threat to your funding sources coming with the new administration, and what we might be able to do about it.
Well, that's a very good question. We don't know where things are heading. I don't think any of us knows where things are heading with the NIH. As far as the new director is concerned, or where it's going to go. I hope that what has been threatened will not actually take place. I believe actually, in earlier administrations, sometimes you've seen a threat, but actually what you've seen is increases in funding. Funding is always an issue. Actually, one of the organizations that I'm a member of, the American Association of Chairs of Departments of Psychiatry, we're on Capitol Hill about a month and a half ago, lobbying our Senator Mark Kelly's office and Kristen Sinema's office to lobby for more and more healthcare dollars for Arizona. And so it's something that we're constantly concerned about. I don't know where things are heading, but we're interested in here at the University of Arizona and ASU and other universities, of course. We are really looking for those of us out here in the community who have the means or are able to donate to philanthropy, where we can have more endowed professors, we can have more seed money for new research, so on and so forth, more funds for buying new equipment, things like that. New research centers. That's kind of where I think, where we would really like to have an influence. It's a lot harder to affect what happens on Capitol Hill, but what can happen in your own hometown, may be more tractable.
Okay, thank you for your answer. And of course, feel free to communicate with ACMI to let us know if we can do anything in terms of lobbying. Obviously, most of us don't have millions of dollars, which if I did, I would definitely contribute to your cause. Thank you.
Thank you. Thank you. Again, speaking of ACMI, I'm very impressed with your organization. Coming from New York City, I was not actually aware of the kind of grassroots organizing that takes place, that is taking place in Arizona at many levels. And it's just really amazing to see, people come together with this sense of common purpose to really, make the world a better place for our patients.
Yeah, I think Josh Moselle, our president, didn't go over just how effective we have been at getting laws enacted. We've had about 21 bills in the past 7 years of our existence, which is good. We're working on more. Again, we're always looking at what bills will actually make a difference with either the data collection, the accountability, the services that we have for people with chronic serious mental illness. I see Jill, you have your hand up first, and Rachel.
Yeah, I'm just wondering, is there much information about young children, say, age 3 younger, showing signs of attention deficit disorder and that ballooning into schizophrenia?
Well, I'll be perfectly honest with you. I'm not myself an expert in. I have seen cases of childhood schizophrenia as young as 5 when I was training at Bellevue. That's a tough question. I'd have to defer that to my child psychiatry experts. We do have a child psychiatry fellowship here at the University of Arizona here in Phoenix. I wish I could give you a good answer. It's just not something I know much about.
Yeah, my son had attention deficit disorder clearly at a very young age. So I just, you know,
Ours too.
so I just wonder about that. And I had heard about in terms of money, and I don't know who, this is just very vague, but there's a family out west in California who owns a vineyard and their son has schizophrenia, and they apparently have given money, a lot of money to that cause, but I don't know. I don't remember which vineyard. Anyhow, thank you very much. This has been really eye-opening. Appreciate it.
Thank you so much. Thank you so much.
Rachel?
Hey, Dr. Fanous, I had to take a dog to the vet and I only caught the tail end, and I cannot wait to watch this just based on the last 10 minutes that I've been able to see. I just wanted to make a quick announcement that Dr. David Taylor, who's one of the leading clozapine researchers in the world, is coming from the UK, and Dr. Deanna Kelly, who is our University of Maryland researcher for clozapine, is coming also next week for the ACNP conference, and we're going to be having a reception. We're having a reception for the angry moms and the mad moms that had the big win at the FDA hearings on the 19th of this month. And so, Dr. Fanous, you're invited. Everybody on this call is invited. I know you're going to be here for that same conference, and that's going to be Monday night. We're going to have a reception. It's a location very close to the conference. And so if you're available, I'm going to go ahead and put the invite information in the chat. Other people on the call, feel free to reach out to me for questions, and you'll see emails about it too. But and thank you.
Well, thank you. Here at the University of Arizona, I mean, we are your allies in this fight. I mean, we are, we're all approaching this in different ways. There's people who are doing the advocacy. There are people who are doing the science, people doing the clinical work. There are people who are doing the education. It all comes together. We're all in the same fight. And I've been very cheered by the sort of the enthusiasm and the support that we've gotten for the science. I see Dr. Chern is on the call. He's been very welcoming in terms of helping us, recruit patients for these studies. I mean, that's actually also a critical need for us. The need to advance research. It's not just the money. I mean, it's what you do with the money. It's like we also have to have outreach and reach the patients who would be recruited for studies like this. They also benefit not just in having hopefully their futures improved by this research, but they also are, we also pay them in cash, and it's a tough time. It's tough out there, especially for these patients. So anything that you guys can do to help us advance our research in terms of recruiting patients. These are not drug trials that we do. Everything that you've seen here, it's not an intervention. It's really just an interview with the patient and a blood draw, and then we do the lab work and things like that. So that's something where I think there are many things that we can sort of do to help each other, and this is definitely something that it really doesn't even cost money.
Yeah. What I can do is I can send out. I'll upload the webinar, and if you send me your slides, I'll wrap them in a PDF and upload them. But I'll send out with that a message to our mail group, which is, we have 800 people on it now, for people that have loved ones with schizophrenia that would like to participate. If you give me how you want them to contact the center or you or whatever, give me that information when you send the slides, and I'll put that in a follow-up note.
Thank you. That would be fantastic.
And a while ago, there was a question about participating in your projects, and I think Jill was the one who asked the question. So I earlier replied that Copa Health clinics can be your storefront. You can have access to voluntary patients, who would be willing to participate.
I'm really grateful to Dr. Chern for opening the doors. Thank you, Dr. Chern. Appreciate that very much.
Yeah, thank you.
Any other questions? If not, we'll let Dr. Fanous go. And if you send me that stuff, Dr. Fanous, I will send that to you. I'll also send you the details of the dinner Monday night. When Rachel provides that, I'll send that out as well.
Wonderful. Again, congratulations on your fine work. Keep up the great work. We're all cheering for you, and we are your allies in the fight. And don't hesitate to get in touch with us if there's anything we can do.
Thank you very much.
Thank you.
Yes, thank you. And we have a lot of patients too at Mad Moms that might be good candidates for trials. So I like this. Cool.
Thank you.
Thank
so
you.
much. From Indiana today, friends.
Oh, nice to have you here, Debbie.
Thank you. I can't always get here, but I could tonight. Thank you.
Thanks. Okay, I'll stop the recording. So if anyone has any requests for help with their family.